This loblolly pine tree growing in the Hatchet Creek floodplain near Gainesville, Florida is impressive. It's large, straight, and apparently disease-free. It has thrived for decades, perhaps a century, through floods, droughts, windstorms, heat waves, cold snaps, and exposure to insects and disease. It looks like the kind of tree you'd want in your planted forest. But how do we know whether this tree's success is due to its superior genetics or just due to the environment in which it grows? This question of genetics versus environment, nature versus nurture, is the central problem of tree improvement. We know that an organism's phenotype is a function of its genotype, or its genetics makeup, and the environment in which it lives. The key to tree breeding is separating the effects of genetics from the effects of environment, so that we can incorporate trees into our planted forests that carry true genetic superiority, the ability to grow fast, with good form and wood properties, and resistance to insects and disease. In this virtual field trip, Dr. Gary Peter and Andrew Sims, tree breeders at the University of Florida School of Forest Resources and Conservation, talked to us about how tree breeders use seed orchards, progeny tests, and quantitative genetics to develop trees with traits suited for establishing productive, sustainable planted forests. Gary Peter starts our tour with a discussion of the selection of superior phenotypes from wild populations.
The largest set of genetic diversity is in the natural stand. And of course, that's where everything started for breeding way back in the 50s. People would go out into the natural forest and look for the biggest tree in a little stand and then select that. At that point, we didn't know anything about the genetics, whether the genetics made it big or whether it was the environment just being rich in nutrients compared to other trees, or it got a faster start or whatever. But those wild selections were really important at the beginning of the breeding program because they created the initial test set, a very, very large test set that then got distilled down to the smaller breeding population we deal with today.
We're standing in what was formerly known as the baseline seed orchard. This is a special type of forest stand that is only useful for producing seed and especially from clones that we're interested in. So by clone, I mean a particular genotype, like one particular person. It could be copied a bunch of times. We copy it by grafting. So all the trees that are in this orchard are grafted, and actually are grafted in multiple places. They were grafted here around the year 2000, and then grafted in the crowns between 2017 and 2019. All of these grafts are partly for us to be able to get genetic material because we need these trees to produce sexual tissues such that they can have the megasporangia and microsporangia conelets that we can get pollen from, and then use that pollen to pollinate the female cones. This is now our fourth cycle / pine breeding orchard. So in this orchard, we have, of course, the genotypes that are here in the main stem, and then we also have several genotypes in the crowns of the trees.
Basically, what the breeding program does is it tries to collect what we think is the best material. It might be the best material that looks like it's growing fast and straight, maybe has small diameter limbs with a certain angle and a good crown form and size. And then we basically try to intermate all of those things that we think are best. We right now look for traits of rapid growth, fungal resistance for fusiform rust and pitch canker, as well as some stem form properties. Those are our main target traits for breeding and in both our populations. And so basically what we do is we take that material of wide variety of genetic types and we try to basically cross them to generate the most combinations possible that we can generate that we think ultimately will be good or lead to better trees that grow faster, have more fungal resistance, maybe and better crown form. And then ultimately we breed those, we generate seed, we take all of that seed out to a genetic test, so also known as a progeny test. And we do that testing at many different sites across the deployment zone. In this case, slash pine is North Florida, South Georgia, all the way out to Alabama, all the way up to South Carolina. And those multiple, we collect the height information, diameter information, whether they have fusiform rust or not, or pitch canker from that field data and from all the sites. And we aggregate all of that. We analyze that by quantitative genetic methods, which are used to calculate something that we call the breeding value, which gives us an estimate or idea of how well that individual does, and the parents. We're really interested in when the parents get crossed, whether they generate a lot of superior offspring. Sometimes those offspring are even superior, and we select those for the next cycle of breeding and then ultimately testing. And so we manage these populations as big. Our current slash pine population has a target breeding population of about 450. We do crosses around 850 to 900 crosses there to generate that 850 to 900 new families that then go out into these genetic tests. And from that, we select for the new breeding population. We try to select again about 400 to 450 of those superior lines. Sometimes their grandparents are still superior. We carry those forward. Parents are still superior. We carry those forward. Other times we select the best progeny of those individuals and carry those forward in our new breeding population for our next cycle. From that set of selections, the sort of 450 new selections plus whatever was old, we can then down select in a much more rigorous way to identify what's considered to be our propagation population. The population will go on in and make its new seed orchard like here. Typically today, that's going to range somewhere from 15 to 25 selections in a seed orchard. So there we're choosing the best of the best, those that have the fastest growth rate with the best disease resistance, so that they can really become the commercial germplasm that's ultimately deployed. And in that way, we can get the best material. We have to screen large numbers to identify those top-ranked families and top-ranked parents to justify putting them into seed orchard. The seed orchards are sort of the deployment, the main way we deploy those new genetically derived families now, by establishing the seed orchards. Seed orchards take a while to mature, so you graft those right selections into the seed orchard, many copies of those. They're going to take typically 7 to 9 years to reach maturity to start producing cones, and then they'll continue to produce cones for 40 years. So really, we seed orchard will have different age scion grafted in it, and they'll be collecting different ages, and they can collect that material both as open pollinated material or closed pollinated or control pollinated material has become very popular as of late. That control pollination not only allows you to choose the father and the mother and know that they combine well in a very general way, their general combining ability works well, but also it prevents some pollen contamination that's present from outside the seed orchard that degrades the genetic value of those individuals, like in an open pollinated scenario. So there's a couple of main advantages of closed pollinated.
So to graft in the top of these trees and to do the breeding work, we need special equipment. Typically we use an aerial man lift like you'd see in a construction site. And for this particular orchard, instead of a 4-wheeled lift, we use a 4-track lift. So the track is to reduce the surface pressure per square inch. We want to distribute that because these trees are very delicate. We're balancing their nutrition. We're taking foliar samples to make sure that they're fertilized correctly. We're really wanting each of these trees to live. because this is, again, a very different type of forest stand than a normal one. So we'll drive the lift into the orchard and go to the crowns of the trees where all the sexual tissues are being generated. And we'll do our work there. So every year it's an annual cycle. As you know, pollen only comes on the pine trees and the females are only available once per year. And so in late December or early January, I'll come into the orchard, go to the top, and see how many of our grafted clones have females that are going to be emerging. Once I see that the female is going to emerge, or a cluster of females will emerge, then I'll put a bag that is air permeable, but it won't let pollen from the outside in. And as the flower develops, it's not a flower, it's a cone, but as the cone develops, eventually the scales will open and it'll be ready for pollination. So in tandem with the inventory of all of the females and bagging them for breeding, I'll also go up and collect males. So males, as you know, come in clusters. We call them catkins, but that isn't proper because they're not flowers. We'll call them catkins. I'll go and collect catkins. I'll wait until they're about ready to shed pollen. Then I'll put them in a craft paper bag. I'll dry them out in the lab, and then I'll sieve it out to where it's just the pollen that's available and dry it down to where it stores well and won't have mold. So once the flower or the female is ready, the female cone, I will get basically a small little air compressing tube like you would see for someone taking blood pressure, and it's hooked to a needle and a tube of pollen. And so I'll blow some pollen around. It's called a cyclone pollinator. So I'll try to circulate the pollen around inside of the bag, seal it back over with tape, and then I'll wait for the cone scales to close. There are six stages of cone development. It's the way it's been characterized. Arbitrary, but it's the way we characterize. And you pollinate at stage 4. So stage 1 is it emerges. 2, you start to see the pink of the cone. 3, the cone's going to start feathering out, and you'll see it as a very small cone, smaller than the tip of your pinky, with the shape of a normal pine cone. And then at stage 4, the scales will begin to open. At stage 5, the scales are open and beginning to close. And at stage 6, the scales have swollen shut such that the pollen tube isn't available anymore. At that point, I'll go back. I'll back up to the top. I'll cut off the things that are holding the bags in place. I'll mark it very well. And then I'll leave it for 18 months because, as you know, those cones need 18 months to look like this and yield seed.
Top grafting is a huge accelerator in our breeding cycle.
As you'll know, young trees don't typically produce cones, either male or female. So one of the ways that we shorten our breeding cycles is to go up to the tops of these trees, 40 to 60 feet, we'll graft, and then the following year we'll look for female cones and male cones.
So in that case now brings us back to the cycle. The cycle of breeding cycle itself now really only takes about 12 years. The initial cycle took 35 years. We're down to about a 12-year cycle where if in an ideal world breeding will only take 4 to 5 years. Our genetic testing will take 4 to 5 years. And then we have 7 to 10 years for after grafting and seed orchard production. And so the full cycle to deployment of new lines takes 20 years. So breeders really have to anticipate and look out into the future. And we really are forced to focus on traits that are general traits that are useful now and will be predicted to be useful there. Growth, volume, those are, disease resistance. That's why we focus on those traits, because those are the ones that pay, and we know that they'll be important, 20 years into the future. So that's part of the constraint. The other element that breeding programs play a very important role in is gene conservation and germplasm conservation. We do that in the context of establishing clone banks. We take all of those selections from our breeding population and we graft those into clone banks at a couple or 3 different sites. So we can go back to that genetic material. So we have a history of maintaining that genetic material in case we need to go back to it at some point, and more importantly, it becomes valuable. The scion becomes valuable for seed orchard establishment, for the top grafting that we use to accelerate breeding now, where we take a scion from a 4-year-old individual, 5-year-old individual, we top graft it, and the very next year it's flowering. We don't have to wait till it gets to be 10 years old. But the wild selections are still really important when we need to increase the size of the breeding program, or if we're looking for new and additional traits to introgress into the breeding population. If we went out, for example, and identified a stand that was very heavily damaged by pitch canker, but we saw some trees survive, those would be good trees to go out and collect and maybe take into our breeding program, because they might have a higher level of pitch canker resistance. And so that would be one way to identify individuals with high pitch canker resistance to introgress into our breeding program, because we really want trees that not only grow fast but have very high levels of pitch canker resistance, because pitch canker resistance can destroy a whole stand. And so being able to identify trees that have high levels of resistance, and then continue to breed those against high level resistance to create even more resistance, we really need to do both. So wild selections still play an important role in helping us maintain genetic diversity, but also finding trees out there that have traits we want. We nowhere near sampled the wild population as extensively as we could have, because we just didn't have that capability.
We are a 8-member agency, or a cooperative group between the Florida Forest Service, the Georgia Forestry Commission, 5 private or publicly owned forest products companies, seedling companies and the like. And the last partner is us, the School of Forest Resources and Conservation at UF.
This research program got started in 1954, and it's been operating ever since. And our focus has always been on breeding southern pines. Started off with slash pine and little longleaf. Now we breed slash pine and loblolly pine, or so-called Florida loblolly pine. The breeding is, and why the cooperative started so long ago, was one of the first cooperatives to ever start in terms of research, is everyone recognized that breeding takes a long time in pine. So overall, we manage a large population, and that takes a lot of work, and it's quite expensive to not just do the breeding and manage the clone banks, but also to do the genetic testing. So that's the real value of working in a cooperative fashion. We work in the cooperative fashion with our members, the state agencies, and the companies, because at the end of the day, we can share that workload, and then everyone gets access to more and better material for deployment, but everyone breeds those different things to share the workload in order to spread the cost out, to enable this whole endeavor, because it is quite lengthy and quite costly. And the role of the university in that cooperative is to manage the breeding effort, to do the selections, the rankings and selections, to make sure that the clone banks are established and maintained, and then also ultimately to do research to accelerate the cycle as much as we can accelerate the cycle. And that's sort of a main set of activities that we do. So at the end of the day, everyone benefits, because the landowner benefits by having improved germplasm deployed that grows faster. That stuff now has to be figured out how to be managed better. The member companies have better germplasm to deploy that gets a better return on faster growth rate. And then the states and nurseries have better material to give to the landowners that can be not just grow faster, but be more disease resistant, that can lead to better stands at the end. Silviculture is clearly an important component in that, but now genetics is really, it being able to advance rapidly, and has advanced rapidly enough now where a lot more of the new advances are going to come back to genetics. We know how to fertilize, we know how to do spacing. A lot of those concepts are out there and have been, very, very important for the increases in productivity. But now we believe that genetics really is going to start to really, compound on that. And that's where the compounding comes in. As we pyramid these better alleles in genes, we get faster growth rate and continue to get faster growth rate. Genetic gains have averaged 10 to 15% per cycle. We're still averaging that, which is around 1% per year, which is about the level of gain that corn breeders get.